present this talk about the logic of containing tumors. So this talk, to begin with, you might start wondering what this has to do with evolutionary rescue, and it's true that that doesn't become apparent to the end, but I promise you there is a connection. So the topic I'm going to talk about is the idea of exploiting competition within tumors as a way of suppressing resistance to therapy. This is an idea that was really first proposed about 11 years ago by these two papers that I've highlighted here, one by Monroe and Gaffney and one by Gatenby and colleagues. And both these papers, which came out around the same time, suggested that by using low doses of therapy, such as chemotherapy, one can exploit competition between treatment-sensitive and resistant cells so as to control tumors, contain them, rather than try to eliminate them. The problem is that if one applies very high doses to try and eliminate a tumor, often there is pre-existing resistance or de novo resistance that emerges, and this leads to relapse, and this is a big problem in the clinic. So colleagues and I tried to test this idea in an experimental system. Well, my colleagues carried out the experimental system and I did some modeling to try and understand what was going on. So the system we had was a human colorectal tumor cell line. My collaborators generated cell lines which were irreversibly resistant to a certain drug. And then they seeded tumor spheroids with a mixture of sensitive and resistant cells. Tumor spheroids are three-dimensional clumps of tumor cells, and they recapitulate many of the properties of a solid tumor, but they're also quite well characterized, which means that they're very suitable for really examining the nature of this competition between the constituent cells. So what I did is I created a computational model that recapitulated the important properties of this experimental system. So in brief, we were able to establish that in the real cells, both in monolayer culture and in mice, the resistant cells paid a fitness cost, which you can see illustrated in the top left of this slide. We also knew from previous work that cells that are farther from the periphery, from the boundary of the tumor spheroid, are less able to proliferate. So I was able to build this property into the model, as you see in the top right. Hypoxia is a very important property of both real tumors and tumor spheroids. And so I created a hybrid cellular automata model which has diffusion of oxygen and effects of hypoxia. And finally, we were able to characterize the effect of the drug on the relative proliferation rate of the sensitive cells. And so each of these components we were able to parameterize very nicely from experimental data. And so the model was therefore quite sophisticated. So here is a result of that model. This is just a two-dimensional cellular automaton, very simple, and it has the properties that I just introduced. Here we will have three models running in parallel. The one on the left has no drug, the one on the right has a very high dose, and the one in the middle has an intermediate dose. And the close-up here shows the resistant cells in pink that are initially randomly located. What we see is that in the absence of any drug, the tumor keeps on growing, and the sensitive cells, because they have a proliferation advantage, they take over, as on the left. On the right-hand side, the high dose of drug eliminates the sensitive cells, but the resistant cells escape, and we end up with a large resistant tumor. In the middle, we arrest the growth of the sensitive cells, and that suppresses the growth of the resistant cells due to competition. And so we're able to contain the tumor and suppress resistance. Now, that's all very nice in a computational model, but did the spheroids actually go the same way? And the nice result is that indeed they did. So here we see the results of the computational model on the left and the actual tumor spheroids on the right. Those growth curves on the right are cut off because we can only grow these tumors to the spheroids to a certain size. So I cut them off before density dependence starts to kick in. And you can see there's pretty good agreement there, which is which is very nice, but it's also kind of expected because of the degree to which we could parameterize this model. So here we found support in an experimental system backed up by this computational model that intermediate doses can indeed be superior in suppressing resistance and controlling tumor growth. And various other people have also made other mathematical and computational models of similar systems. You see a partial list here. The paper I was involved in was Bacevich and Noble et al. 2017, somewhere in the middle. And you can see there's been a lot of work here over recent years. But a colleague and I decided to try to see if we could generalize these previous models, because as you can see by the competition type here and the methods, all of these are really very particular models. And we wanted to establish whether there was some general class of models which has the same properties that have been established in these particular cases. So I teamed up with the mathematician Yannick Vieussach, who's based in Paris. And what Yannick really developed is a very general model here, where we have two types of cells. We have sensitive and resistant, S and R. And we have a very general growth rate function, lowercase g here, which can be different for the sensitive and resistant cells. And this is a function of the sensitive and resistant population sizes as they change over time. And the sensitive cell growth rate is also a function of the drug dose, C. And we make a couple of important assumptions, which are supported by the biology. The first one is that the growth rate of the sensitive cells is non-increasing in the density of resistant cells and strictly decreasing in the drug dose C. So this just means that there's competition, and there's also some effect of the drug. The drug has a negative effect. And then we assume that the growth rate of the resistant cells is strictly decreasing in S. In other words, the resistant cells suffer from competition with the sensitive cells. These are the key assumptions. We make a few other technical assumptions, but these are the most important ones. And what Yannick was able to prove very generally and very rigorously is that under these assumptions, two important results hold very generally. So the first one is that the optimal strategy maintains tumor burden as high as possible as long as there are some sensitive cells left. We call this ideal containment strategy. And the worst option is actually to maximize the kill rate. This is the maximum tolerated dose, which is actually the default strategy that's often used in the clinic, trying to eliminate tumors. What we show is that if the assumptions on the previous slide hold, and if there is pre-existing resistance, then that's actually a very bad strategy for prolonging survival. However, although that is qualitatively true, quantitatively, if we look at the size of the clinical benefit, that really depends on the nature of this competition between the cells. So here we look at three different kinds of density dependence, which have been used in modeling density dependent populations. And on the left there, you see three growth curves for these three kinds of density dependence. And you can see that those growth curves are rather similar to each other. We've created them so that they resemble each other. And on the basis of some tumor data, it might be hard to tell which of these is the true model. However, if we look on the right and we see the relative benefit of this containment approach as opposed to maximum tolerated dose, then we see that there's a very large difference between the different model predictions. So if the nature of the competition is logistic, then the containment approach is not much better than maximum tolerated dose. If it's von Bertalanffy, then there's a very big difference. And Gompertzian, which is a very standard model, is somewhere in the middle. So in our paper, we present results for various models, but we particularly focus on the Gompertzian model, which, as I say, is a very conventional model in oncology. And so here is an example of a Gompertzian model. Here the competition is contained in this term, this log of k divided by n. So k here is a carrying capacity. And we just have a simple function here on the right of the first equation, which shows us the effect of the drug. And this model was previously studied by Monroe and Gaffney back in 2009, but we were able to obtain analytical results, whereas their results were mostly numerical. And what we were able to show is that for this Gimpertzian model, we can get analytical results for the advantage of the containment strategy versus maximum tolerated dose, both in time to progression and time to treatment failure, and also overall survival, which isn't shown here. So we can get these very nice analytical predictions that are based on the parameters of the model. And there are a lot of these kind of predictions in our paper. So previous papers have suggested that an essential component in this containment approach is that resistant cells must pay a fitness cost of resistance. What we were able to show is that, in fact, this isn't necessary. All of the results you've seen so far haven't assumed that resistant cells are generally less fit than sensitive cells. However, we do find that a fitness cost can be very, very beneficial in terms of the size of the clinical benefit. So here we consider an extension of the previous model in which we're able to model different kinds of resistant costs. And I list there 3 different ways in which there could be a fitness cost of resistance. In particular, you can see here that inability to compete with other cells is an important form of resistance. So if we examine that factor, then we have on the 2 axes of this heat map, we have 2 forms of competition parameter. And what we see is that if both of these, if resistant cells are poorly able to compete both with sensitive cells and in general, so they have high density dependence and they're particularly poor competing with sensitive cells, then we find that the clinical benefits of the containment approach can be very large. What we see here is the relative benefit. And in fact, if these parameters are extreme enough, then the benefit becomes infinite. There's a discontinuity here. And essentially, in theory, the patient could be kept alive forever by this kind of approach. And we're also able to look at different eventual outcomes. So what I looked at there is the relative survival, the ratio of survival under containment versus survival under maximum tolerated dose. What we look at here is what eventually happens. And the green region here corresponds to the green region in the previous plot that shows where the tumor is in fact tolerable. And there are different scenarios depending on these parameters.